Hello everyone, welcome to my channel. Today we will be discussing about dark field microscopy. So we will see the working principle of dark field microscopy, the various applications, advantages and also the disadvantages are associated with this particular type of technique. So to start with, first the working principle. Now, if you can see this images, one here and the other one here, they have one uniqueness. Their background is completely dark and then you can see the sample which is brighter. So in both the cases, you see the sample is much brighter and the background is comparatively very dark. And from this, the dark field microscopy or the name has come. Okay, so the entire field, the background specially remains dark, and that is why the name is dark field microscopy. Now, let's see how the working principle or on what principle this actually dark field microscopy works. Okay, so basically, if you can see on my screen, these are the condensers. Okay, they are specially designed to work with dark field microscopy. But before coming to the condenser and what is special about them, we have to understand what is special in the light path or how the light path is diverted so that you get a contrast. Now, what is contrast? As you saw in the previous image, your background was completely black and your sample was brighter. So that is the contrast you are generating without adding any external dye and thus getting a brighter image in a black background. So that is the contrast. Now, coming to the point, how do we get that contrast, right? If you see this light path, in this light path, so this is the light source. The light source is coming from here. And in the light source, if you can move ahead, then in this moving ahead path, you have the first point, or if you can see the biggest block is basically the condenser. So which is showed here. Okay, so these are the two types of condensers which I have been talking about, and the design I'll come to it in a bit. So the condenser's role is to condense all the rays of light onto your sample. So this is the sample where the sample is being kept. The condenser will focus all the light, condense all the light passing through it onto the sample, so the sample is very brightly illuminated and also evenly illuminated, right? So that is how the name of the condenser has come. Now in case of a dark field microscopy, what is happening, you see your rays of light are coming in all directions from the light source. So you see the rays of light coming in all directions, okay, as you can see here, and also this path. What is happening here, there is a block in between. So this block is basically restricting the light coming within this region to pass through it. Okay, so what actually is happening, this entire region of the light path is being blocked in the center, and only the light in this, you can see the yellow one and the dotted one, the light in this is allowed to pass through it. So if I draw it through blue lines, your light is basically allowed to pass from here and light is allowed to pass from the other side. Okay, so what is happening in the center, basically if you see, this is like a circle. Okay, so if this is a circle and your light eventually was passing all through it, correct, in the previous case, but in the dark field, what is happening, there is another ring here and this ring is actually blocking your light to pass through it. So if you can see here, this is what I have been talking about. This is the ring, or rather this is the ring which is passing or which is positioned in the center of the condenser, and it will block the light. So you can see light can only pass through the periphery through these gaps only. Okay, so in similarly through this gap light can only pass, and this light is allowed to fall onto your sample. Till here it is okay. So you have light, you have a block in the center. The blocks are in the form of this. You can see the blockage in the middle, and you are having the light focused onto your sample from the periphery, from the edges. Right. Now, what is happening if, suppose, example, this ray of light doesn't no come in the in conjunction with the sample. So if it doesn't passes through the sample, there will be no deviation in the light path and it will go straight. Correct? Similar thing is going to happen for this side also, and it will go straight without facing any deviation in the light path. Now, why this deviation will come? Because there's a phenomena called diffraction. So if you have missed what diffraction is, I think you can check my previous video where I have discussed about diffraction in details, right? So here, if you see, if the sample, or if the sample is in the light path, then what is happening? Because it is passing through the sample, the ray of light is getting deviated. Okay, and this deviation or the change in light path is allowing the light to be collected only from this region, in the inside region, not from the outer side region. Okay, so whatever light is actually passing through the sample, that particular ray of light is bending towards the normal. So it is bending towards the normal, towards the inner side, and because of that, what is happening is it's bending towards the normal, and that is why you are getting a diffracted light in the center. So this diffracted light is diffracted by the specimen, and only the light which are being diffracted by the specimen are collected or used to create dark field image, and that is why your sample looks very bright, because the light only passing through the sample is being collected, and your background looks absolutely dark because that light is not at all collected. So this particular light, which is not deviated and does not pass through the sample, it does not change the light path, it goes straight and it is not being collected. Okay, so if I explain with another ray diagram here with different working distance of the objective. So if you can see here in the first case scenario, this is the condenser. The light in the center is being blocked from this region, and you are getting collecting only light from the periphery, from the edges, and after diffracted, you see the light is collected by the objective. So objective is collecting only from this region. This light is not collected, this light is also not collected, which is from the background. And if we decrease this length, then there is a chance of this light is also being collected, and that is why we see a different type of objective here, which are specially designed for low working distance or small working distance. So we say this is W and D, that is the working distance. Basically, in other term, that is the focal length. So sample, and this is the objective. The focal length of this particular objective is the working distance. Now you see the light is again collected from the center. Also, because the working distance is less, you're also collecting rays of light from the background, but this light should not be allowed to form an image, and that is why there is a iris diaphragm, and this iris diaphragm blocks the light which are coming from the background. So that is the mechanism how you block the background light and you only allow the image to be formed from the light which are actually being deviated from the sample after passing through the sample. Okay, so in comparison with a bright field, how does a dark field stand out? So if you see here, this is a ray diagram for the bright field, and this is a ray diagram for the dark field. So very simple example, and how you differentiate them by, in case of a the condenser lens, in case of a bright field, you see the light is collected from the entire lens. There are no restrictions. Whereas in case of a dark field, you have the central aperture, which will stop the light in the center. So it will light will be only from the edges. And because of this, we get a scattering effect. And this scattering effect will only occur if the ray of light is passing through the sample, which will have a brighter light, which is the diffracted light from the sample, and your background light is not collected. In case of a bright field, this does not happen. Whatever light is being condensed, all the rays of light passes straight, and you get some signal from the background and some signal from the sample. Okay, now in case of a microscope architecture, if you know this architecture, I think this looks familiar to most of you. So the binocular, or this is the eyepiece, you have the objectives, you have the stage. So in this case, where you have the condenser in the below, in the condenser you have to put the central aperture, which will go and sit here, and then you can collect your dark field image, right? So apart from that, there are no difference. It is basically the same light path. Okay, now what bright field applications can be, why are we using this bright field, and how we capitalize this technique to get a benefit out of it. The first and simple thing is, if I can show you here, this is basically like diatoms. So they are so small, you don't need to stain them, or you cannot even stain them. So you just have to put them on the slide, and you can do a beautiful dark field image. As you can see here, they are such tiny, but their architectures are so good, and then you can easily see them. So if I zoom a certain portion, you can see the internal architecture, and none of them are stained. Similarly, if you see bacteria, so in case of a bacteria also, you see the bacteria are so small, still you can see them very distinctly against the background. Okay, and in case of samples, like you have the larva, which is a mosquito larva here. In this also you see these edges, they are the legs. In case of a cilia also, you do not have to stain them. There is no point of staining them, and there are also conditions where staining is difficult. So in all those type of samples where you cannot stain them, you just want to put them on the slide and see it in a dark field, yes, you can go ahead. And what basically, this is like, these are the examples I have picked up for you for your reference, like a mosquito larva, bacteria samples, diatoms. You can have like RBC cells. In RBC also you cannot stain them, and you just want to see the morphology of the RBC. And also for material sciences people, like you have some edges, you have grains, you have scratches at the edge of it, and you have small crystals, you have silica surfaces. Okay, and then you want to see at the edge whether there are cracks or defects. In all these studies, you have to know, think like how you can prepare. But in case of a dark field, you don't have to think how you prepare your samples. You just put them on your glass slide and you can image them beautifully. Okay, so advantages, yes, you don't need any sort of staining, just the sample as it is, you can put it, and then are good to get the images, best images. Okay, samples with similar refractive indices are better. If the refractive index changes, then we may have problems, but usually samples with similar refractive indexes are preferred. And as I mentioned, for like material sciences people also, like you can examine defects. It can be the edges of it, the grains of it, and these defect studies are very easy, as you don't need any time invested on preparing a sample before getting the images. But yes, there are also certain disadvantages associated with this type of microscopy. That is, there are cases of image distortions. Image distortions usually when you have the sample, are in multiple layers, you do not get a sharp image. If your sample thickness is also varying over the entire field of view, then also you have artifacts and image distortions. One very common problem with dark field microscopy is dust. In India, we know there are a lot of dust. It is very difficult to maintain a dust-free lab. Yes, people do, but it is a very, what you say, a day-to-day task. You have to maintain the microscope in a dust-free environment. But if there are dust, what will happen? This dust, when comes in the light path, they will diffract the light, and you will have lot of false positive signals in your image. So you will see brighter dots in your image, which are basically contributed by the dust in the light path. And lastly, it is dark field microscopy technique is not suitable for oil or water immersions. The one basic reason for that is you cannot avoid bubbles. So the bubbles or the edges of the bubbles will cause lot of diffraction of the light, and your entire objective of getting a sharp image against a dark background is lost. Okay, so next time when you're thinking of a dark field microscopy, think of the advantages and also the disadvantages. What type of sample you're using, whether these samples are suitable or not. All this you please think, and then you decide for the technique of microscopy which to be used for your imaging purpose. Okay, so these are some of the references. You can use these references for further studies. Thank you.